Hello, in this module I'm going to look at the first part of the structure of DNA. So let us look at the structure of DNA. Now what you see here basically is an image, okay? And this image is nothing but it was based on X-ray crystallography provided by Maurice Wilkins and Rosalind Franklin. Fine. Based on this X-ray image, two scientists, namely James Watson and Francis Crick, proposed the helical structure of DNA. That's James Watson and Francis Crick, and this is their actual model of the DNA. Fine. So once again, we'll see that Maurice Wilkins and Rosalind Franklin in the Feb of 1953 basically proposed the X-ray diffraction pattern of DNA, based on which two scientists, James Watson and Francis Crick, actually proposed the helical structure of DNA in the March of 1953. Same year, just a month later. Okay, let us see this structure of DNA and let us learn that in points. Okay, so we're looking now at the structure of DNA and we're going to analyze the DNA structure in these 7 points. Starting with the first point, DNA is a double helix. Now why it is called a double helix? That's the image right here. So what you see basically here is it consists of two strands coiled around a common imaginary central axis, okay? And that is what makes a double helix. Correct? Now if you look at this image, it actually appears like a twisted ladder. So we can also say that DNA looks like a twisted ladder. And if you see here, you'll also see alternating major groove, minor groove, major groove, minor groove, major groove, and minor groove. They alternate each other. So the first point is DNA is a double helix. Okay, now let's look at next point, which is structure of each strand. Now if you look at a single strand of DNA, this is what it looks like. It is nothing but a long chain of nucleotides. Let's recollect what was a nucleotide. Now each nucleotide is further made up of deoxyribose sugar, because it is DNA. It is made up of phosphate group, right, provided by phosphoric acid, and it is also made up of one of the 4 nitrogen bases. You can see all 4 here, A, C, T, and the one that is labeled is G. Okay, fine. Also remember that the 2 successive nucleotides of the same strand are linked by a special bond which is called as 3'5' phosphodiester linkage. Why is it called that? Because the phosphate groups basically forms ester bonds with the 5th carbon of one nucleotide and the 3rd carbon of the another nucleotide. And this is called as the 3'5' phosphodiester linkage, or you can also call as the phosphodiester bond. It is a strong covalent bond that links the 2 nucleotides in the same strand of the DNA molecule. Okay, moving on. Let us look at why they are called as complementary base pairing. Okay, now in a double-stranded DNA, what you see is purine will always pair with pyrimidine. Always. Okay, to be more specific, adenine, which is a purine, will always pair with thymine, which is a pyrimidine. And between the 2, you will see there are 2 hydrogen bonds that connect the 2. Okay, then to be more specific, guanine always pairs with cytosine, okay, and vice versa, which means G and C are always bonding with each other, and between them you will see 3 hydrogen bonds. Okay, moving on. Now let us consider the base sequence following the DNA strand. Okay, correct? What you see here is a DNA strand with the base sequence A, C, G, A, and T. Now let us see if you can predict the base sequence of the other strand. Let's see if you got it correct. Obviously, with A you will see T, with C you will see G, with G you will see C, with A you would see T, and with T you would see A, because they are complementary bases. They complement each other. Now let's add the H bonds. Between A and T, you see the 2 H bonds, and between G and C, you see the 3 H bonds. Is that okay? So what you can say here is, since the base sequence on one strand of DNA decides the base sequence on the other strands, the 2 strands are called as complementary strands. Now, if you look at the nitrogen base along with their hydrogen bonds, right, together they actually look like rungs of a ladder. Okay, and they're also called as transverse bars. and the distance between two successive base pair is about 0.3 nanometers. Okay, yes, fine. Moving on, let's look at the purine to pyrimidine ratio. Point number 4. In 1940, a scientist called Erwin Chargaff, right, he gave the pattern in the amounts of the 4 bases in a double-stranded DNA. Fine. Now, according to him, complementary base pairing, total number of purines is always equal to total number of pyrimidines. And this is also called as the Chargaff's rule, right? Because A is always equal to T and G is always equal to C. Or we could say that purine is always equal to pyrimidine. Okay? Now, let's write it in another form. You can see here. Now, if purines is equal to pyrimidines, you can also say that A plus G, which are purines, will be equal to T plus C, which are pyrimidines. Or ratio of A plus G, which is T plus C, will always be equal to 1. Correct? Because purines are always equal to pyrimidines. And this is called as the Chargaff's rule. Okay? Now, in this module, we've talked about the first part of the structure of DNA. In the next module, we'll look at the remaining part of the structure of DNA. Thank you.